welcome. So this is the second of 3 webinars in our BioGeoSCAPES Modeling Webinar, and I'd like to welcome you. And just before we get started with the main subject of this webinar, namely 2 talks, just a few introductory words on the program that we're about and about this, the purpose of all this. And so maybe we can just start. So maybe a few words about BioGeoSCAPES, because it's in the name. So BioGeoSCAPES is a new global research initiative, and we've launched it to advance understanding of ocean microbial biogeochemistry. So we want in the end to develop predictive models, and we also want to promote international collaboration. And there are basically 3 pillars. We want to map key ocean metabolism over space and time. So we want to get a real understanding and maps basically of the abundance of key microbes and where are which microbial biogeochemical processes happening, what's driving them. Then we want to understand the mechanisms that connect these different metabolisms and the productivity in biogeochemical ocean models. And well, we want to build a conceptual framework to understanding the links between this ecology of microbes and biogeochemical cycles. And then we want to come to something that allows us some predictive modeling. So we want to predict interactions with environmental factors to provide assessments of what are possible risks, what are vulnerabilities in the key ocean metabolisms on a changing planet. So that's the plan of BioGeoSCAPES. And for that, there are basically 3 very different scales that we look on. So one important part in our planned increase of understanding is to understand intercellular processes. So, and for all that, there are very different models are available. And then we want to go to the larger scale also to understand organisms and assemblages. So we want to get to the scale of the ecosystem and not just single organisms. And then we also want to get to the large scale picture. How does that affect ecosystem scale cycling, biogeochemical cycles and feedbacks? And to do that, we want to connect the modelers that use very different types of models. So on all these scales and different techniques. And we want to highlight what's available, the diversity of models, and assess how well can they contribute to the goals of BioGeoSCAPES. And also with this webinar, we want to initiate collaborations between modeling groups within BioGeoSCAPES, and hopefully we'll come to developments in models from there. So that's the goals of the modeling workshop. And basically we have 3 modeling workshops. One has already happened last week, was on the large scale biogeochemical models. Today is more on the ecosystem side and will deal with network and statistical approaches. So our speakers today will be Corday, Selden and Lionel, Guidi. I'll say something on them on the next slide. And then next week we have a final webinar on the cellular scales, and at the same time, everyone who has registered for one of the meetings is registered for all of them. So we invite you to look at all of them. They will also be recorded, and the recordings will be made available. And then in the end, we aim to bring people together at an in-person workshop, which will happen in September at Woods Hole Oceanographic Institute, so 2 hours away from Boston. And the applications for that will open next week, basically. And everyone who is participating in this webinar will get getting an email concerning that. Okay, so today's speakers, we have basically Corday Selden, who's currently a postdoctoral scholar, but soon to be assistant professor in the Department of Marine Coastal Science at Rutgers University. And Then after that, we will have a talk by Lionel Guidi, who's at Le Laboratoire d'Oceanographie de Villefranche, CNRS in France, and who's also strongly involved with Tara Oceans. And our plan is that now these two will give 20-minute presentations, and then we will have a discussion session afterwards. So you will have on your screen a Q&A button where you can basically submit questions. And in the end, we will basically try to get through them. Let's see how far we get and get a moderated discussion going. And finally, just an important thing in this type of things is we have a code of conduct. We do not, in our seminars and also in all the things that we try to organize, do not tolerate harassment or bullying and discrimination. We would not like to have physical or verbal abuse in this type of meetings. And we would also not like to have any disruption of presentations or other forms of communication. So please be all respectful. We can criticize, but we can critique ideas, but not individuals. If you observe something that is not does not fit with this code of conduct, please be an ally. So report it. This can be difficult, I know, but nevertheless, I think it's good to do it. And scientists can discuss pretty hotly, but be patient and kind. So with that, I'd like to invite Corday, who's the first speaker, to share her screen and do her presentation. Thank you.
All right. Thank you so much for that introduction. So it's an absolute pleasure to be with you all here today. As Christoph mentioned, I am not a numerical modeler by trade. That said, many machine learning tools are really approachable for those of us who lack a background in mathematics, and I've spent some time in the past few years exploring their value in augmenting my field and lab research on ocean metabolism. So today I'm going to share with you a first foray into this space, a paper from my PhD, which applied a supervised regression analysis approach to interpolate low resolution but very high effort measurements through time and space. Second, I'll discuss some ongoing work in which I, in collaboration with a few colleagues and students here at Rutgers, have been applying the recent explosion of new machine learning-driven tools developed for applications in the field of structural biology to elucidate micrometal interactions in the ocean. Now, we know that microbial processes are shaped by rapid shifts in local chemistry and physics, necessitating observation at fine temporal and spatial scales. But many of our direct biogeochemical measurements are laborious and expensive, and this can often make it difficult to achieve high spatial and temporal resolution. If we're measuring the rate of a metabolic process or elemental stoichiometry, for example, we typically will only make a few point measurements through the water column per station, and our stations may be pretty far apart. Consequently, we can miss episodic biogeochemical features like this purple hotspot I'm showing at the front in this image. And if we're integrating measurements through time and space, we may end up under or overestimating masses or flux rates, and by extension of the latter, potentially geochemical budgets. We may also struggle to assess environmental drivers of microbial metabolism due to scale mismatch and small sample sizes, which can limit the statistical tools we can apply during data analysis. Now, aside from sensor and geochemical method development, both very important, this type of problem is at its root an issue of how to most effectively interpolate measurements through time and space, and is highly conducive to classical supervised machine learning approaches. Here, when I say supervised, I mean that a model is trained on a labeled dataset, that is, a dataset that includes both the input data and the correct output or target for each example. The goal is for the model to map the connections between inputs and outputs so it can make accurate predictions on new, unseen input data. This approach can be classification-based if we're working with categorical data, or regression-based if we're working with continuous data. Very briefly, we'll typically start by reserving some of our data from training, which we'll use to test the model later. We then will generally split off a subset of the training data for validation. The remainder is used to train a model. Now, there are a lot of different types of algorithms we can use here. I'm not going to go into any specifics. The model trains by iteratively adjusting its internal parameters, like weights in a linear regression or splits in a decision tree, to minimize prediction error. Once the parameters have been optimized, we evaluate model performance using the validation dataset that we've reserved. Based on these results, we can then tune hyperparameters. These are things like maximum tree depth in a random forest model, and retrain our model. Once the model is optimized, we can then assess its performance using the reserved test data that was not involved in training. Now, while this form of exercise is becoming increasingly common in biogeochemistry, there's some really important limitations to consider. One of the most important is the potential for over or underfitting the model. Underfitting is when the model is too simple to capture the underlying structure of the data. This can occur if you lack suitable input variables. Overfitting is when the model learns the training data too well, including noise and random fluctuations, and fails to generalize to new data. These issues of fit can mean that your data coverage is typically the single most important factor in determining how broadly you can apply your model. Attempting to apply a model outside of the boundaries of the training and the test data can lead to significant errors. So I'm going to show you an example of this type of classical machine learning model. I first started playing around with this sort of approach as a graduate student working on nitrogen fixation. And in particular, I was interested in the contribution of nitrogen fixation on continental shelves to the ocean's nitrogen reservoir. Now, coastal diazotrophs have long been discounted as significant contributors of new fixed nitrogen. However, just prior to the study I'm going to share, our group had compiled a strong interseasonal data set and estimated that an area of the mid-Atlantic bight, right, that's this part of the shelf I'm showing just, you might recognize Massachusetts and Woods Hole up there just on the northern part of the western North Atlantic shelf. And we found that an area of the mid-Atlantic bight representing only 6% of the North Atlantic continental area fixed as much new nitrogen as had been previously estimated for the entire basin's shelf area. So clearly there's a lot more nitrogen fixation happening on at least this shelf than previously thought. I then had the opportunity to measure nitrogen fixation rates on a cruise spanning this really interesting physically dynamic region, the cruise track for which I'm showing in this plot on the left. And this is where southward flowing waters of the mid-Atlantic bight coastal zone and the South Atlantic bight coastal waters converge along with the northward flowing Gulf Stream and ultimately diverge from the coast. I measured some really exceptionally high nitrogen fixation rates on this cruise. I'm showing you just the surface data here using a log scale. Now, values on the order of 10 to the first nanomoles of nitrogen per liter per day would typically be considered pretty high in most areas. And here in the red box, we saw values on the order of 10 to the second, which are among the top 5% highest nitrogen fixation rates measured globally. Now, given that these rates were observed in a dynamic frontal zone, I was curious to get some insight into the role of regional physics in driving this hotspot. So I built a few different machine learning models to predict nitrogen fixation rates from available hydrographic data using my direct measurements as the training data set. This was a moderately sized data set, around 200 data points or so, and I ultimately settled on a random forest algorithm. I built 3 different versions of the model using different hydrographic data as input. Here, I'm just briefly going to share what I saw using a combination of latitude, longitude, depth, mode of sea surface temperature, and chlorophyll A fluorescence as our predictors. Before we move off this slide, though, I want to note a few things. First, while all 3 models performed pretty well, with R-squareds higher than about 0.8, they tended to overestimate low values and underestimate high values. And this is typical of this of random forest models. There's also some major outliers, and this is particularly true for Model 3, which included fewer explanatory variables than the other models. So this is that issue of underfitting. Now, looking from a top-down view, we can see the model predicts high rates near the coastline and offshore north of Cape Hatteras, near the edge of the sloped sea. Taking those same predictions from the surface waters based on satellite data, looking at them with seafloor depth on the x-axis. So here the left side of the plot is right snug up next to the coast. The right side of the plot is about halfway across that broad continental shelf. We can see these patterns more clearly. Here we can see that the convergence, that both north and south of the convergence zone, nitrogen fixation rates spike just offshore as chlorophyll concentrations begin to drop. And this is likely as terrestrially sourced nitrogen is being drawn down, but other nutrients remain available. Zooming out a little bit to include the slope, we can see another predicted nitrogen fixation hotspot as mid-Atlantic bight shelf waters mix with Gulf Stream waters as they push offshore. Now, speculation on the how and why here are beyond the scope of this talk, but this modeling work offered us more fertile grounds for further hypothesis development, which ultimately led to further work, a direct observational study on the role of frontal mixing in stimulating diisotrophy. And if you're interested in learning more about that, check out my JGR paper. The reference is at the top. Now, I'm going to move on to discuss how some of these machine learning approaches are driving new fundamental knowledge in the field of structural biology, and how I think we can start leveraging some of those advances in marine biogeochemistry. The first really major scientific problem to be solved by machine learning is what's known as the protein folding problem, a challenge posed by John Kirchhoff during his 1962 Nobel acceptance speech. Proteins are polymers of amino acids, and their 3D structure is, in theory, a function of their 1D amino acid sequence. Yet predicting their 3D structure has proven exceptionally difficult. It took until 2021 for this challenge to be declared solved. And this was by an AI algorithm called AlphaFold, which was developed by Google's DeepMind group based on foundational work from David Baker's lab at UW. And in fact, David Baker took 50% of the Nobel Prize. Now, the AlphaFold architecture is pretty complex. In brief, it's composed of 3 discrete functions. When an input sequence of amino acid residues is entered, AlphaFold conducts a database search and extracts similar sequences from a diversity of organisms, generating a multiple sequence alignment. It also conducts a pair alignment and searches the PDB for proteins with similar sequences and an experimentally resolved structure. This is then used to create an initial pair representation of the input sequence, representing the relationship between every pair of residues within the target protein. This data is then fed to a neural network with 2 towers or parts, one that evaluates the importance of each residue in the sequence, and the other that evaluates relationships between residues. These 2 functions are able to cross-communicate iteratively. They do this 48 times before arriving at a final refined MSA and pair representation. These representations are then fed into another neural network called the structure module, which gives an initial guess for a 3D structure. It also applies fundamental physical and chemical constraints dictated by atomic bonds and angles, right? So things like torsional stress. This guess is then cycled back through the first neural network and used to refine the model prediction. Right? So this is a very computationally expensive approach, right? But one that was necessary to solve this problem. The training data set for AlphaFold, and in fact for all other protein models, is the protein data bank, which is housed at my home institution of Rutgers. The protein data bank, which I'll refer to as the PDB, contains all experimentally derived protein structures, now at roughly 235,000, up from 7 when it was founded in 1971. As any crystallographer will tell you, these structures are really laborious to produce, right? They can take years to perfect the crystallization approach for a single structure. And consequently, the PDB represents only a tiny fraction of the protein diversity found in nature. It's also skewed towards more easily crystallizable groups, and unsurprisingly, those of relevance to human biology and medicine, particularly from model organisms like E. coli and yeast, but also from a lot of mammals. Now, at this point, you might be asking, what value can predicting protein structure bring back to biogeochemistry? Well, a particular interest of mine, and I think of many of yours, is in microbial metal utilization and cycling. The primary role of transition metals in a cell is to act as protein cofactors, offering stability, catalyzing chemistry, and coordinating substrates. I've consequently been thinking a lot about how we can start using ocean omics data to say something about metal demand, and really what I mean is metalloprotein expression, intracellular metal ligand complexation, potentially, and maybe by extension, I'll get to this at the end just very briefly, extracellular metal ligand complexation. Now, the groundbreaking version of AlphaFold that came out in 2021 did not include prediction of protein cofactors. However, at the time and in the decade preceding this, there were many models coming out which used more traditional AI approaches, similar to more complex, but somewhat similar to what I described at the beginning, without a structural prediction to predict metal cofactor presence for a given sequence. I'm going to discuss one which was developed by some collaborators of mine here at Rutgers called MeBe-Pred. Now, following AlphaFold, as you might expect, a slew of additional models sought to predict cofactors for AlphaFold predicted structures. I'm going to discuss one called AlphaFill briefly, which uses a sequence and structure comparison to furnish AlphaFold structures with cofactors. There have been literally dozens published in the past couple of years, including many others that use AI approaches. Now, more recently, AlphaFold 3, which was released a few months ago, now also includes metal cofactor predictions. These AI approaches to predict simple cofactor presence have the advantage of being less computationally expensive and thus much faster. So we're going to take a look first at what they can do for us. Before we begin to look at ocean samples, it's prudent to begin by examining well-characterized proteomes, things we know. On the left, we're looking at MeBe-Pred predictions for a range of high-quality proteomes available on Uniprot for upper ocean organisms. On the right, we're looking at culturing field data showing metal abundance in phytoplankton and a surface particulate pool normalized to phosphorus. Now, we can see significant variability among our marine microbes. The bars here indicate standard deviation among a couple dozen different high-quality proteomes that I pulled. Right? And we can see that they vary in the proportion of their proteomes predicted to be particularly calcium and potassium binding, both of which make up a pretty substantial fraction of the proteome. Iron proteins predicted to make up are predicted to make up only about 7% of the proteomes of our marine organisms, and this is less variable across taxa. Right, so we can begin to get some insights here, and this approach can potentially provide new insight on the metal dependence of different metabolic functions and the metal requirements of different organisms, and consequently, could be really valuable in complementing direct biogeochemical approaches like those that I'm showing in the plot on the right. But before we get too invested in a potentially complementary tool, we want to try to assess its accuracy. Now, this is a difficult task. The accuracy of the model is reported, right, in the publication, but it's reported, much as I described at the beginning, based on its performance on test data subsetted during training. But our applications might look quite a bit different than training data derived from the PDB, right? We're dealing with underrepresented taxa, and ultimately we want to be able to apply these approaches to omics data from largely uncultivated organisms. So as a first pass, we can assess the frequency of true positives by comparing cofactor predictions to annotations in Uniprot. Many real metalloproteins may not be annotated in Uniprot, so we can't really get at the frequency of false positives this way, but at least we can get a first look. Now, I'm going to show data from two microbes, Thalassia cyroproteum and a Prochlorococcus. This plot is the same as the one that I showed you a moment ago, but I'm very sorry, I've reorganized the x-axis and it's alphabetized now. Here, we're looking at the percent of true positives out of the total proteins annotated as binding each metal shown. Off the bat, we can see some troublesome variability with less than half of the known iron and zinc proteins being correctly identified here. Though more power consuming, we can take a similar approach to look at results from alpha-fill furnished alpha-fold structures. Here, we can see a much smaller proportion of the proteome is predicted to be metalloproteins with fewer iron proteins and more zinc proteins. But the structural approach makes our rate of true positives much higher for both organisms. This is above 97% for all metals examined, right? So this is much more encouraging. Again, we're just looking at true positives. We don't know the rate of false negatives, but it does put us on track. Now, in addition to improving accuracy of the occurrence of metals in a protein, the structural approach provides greater information, right? Particularly how a metal is bound within a protein. How a metal is bound in a cell is important because whether it is held by stronger or weaker ligands likely determines its bioavailability and its reactivity during organic matter regeneration. Now, in my opinion, application of these new machine learning tools to predict metal cofactors and metal binding site configuration from ocean omics data is currently limited by the availability of sufficient validation data sets. So this is a gap that I've been working on addressing with some of my colleagues here at Rutgers. But ultimately, it's going to require a larger scale community investment. So if you're interested in having a chat about this, reach out to me or come find me at the workshop. And with that, I'm at the end of my time. I will thank you very much and leave you with these takeaway messages.
Thank you, Corday. When we leave, yep, okay. Then I think we just proceed to the next speaker, Lionel Guidi, and have a discussion at the end, just to make sure we stay in time.
I guess you're talking, Christoph.
Sorry. I just wanted to remind people that there is this Q&A button where people can put questions for the discussion. Just thank you. Sorry, and go ahead, Lionel.
Thanks. So thanks a lot for the invitation. It's a great pleasure to be here. It's a great community, and actually the workshop has a lot of interest to me because I've been trying to tackle this question for a long time, and with few colleagues, wrap my head around trying to understand the biological carbon pump. And I've been studying the biological carbon pump for about 20 years before the omics data were here and before the imaging data were here as well. And so I'll try to take you through a journey of maybe the last 10 years, maybe not the last 20 years, of what was my thought around it and how we moved from getting very few data and a low understanding of the biological carbon pump to something that now is more getting into depths of the biology itself and how the biology is driving the process. So for those of you that are not familiar with the biological carbon pump, we should be only few of you, I guess. But to me, it's always been defined as a very simple process. I mean, it's a complex process that has been defined with simple principles. So basically, the total biological carbon pump is driven by three major processes: the primary production, the carbon export or carbon flux at different depths, and remineralization. All these processes are actually driven by biology and are very complex when you get into the depths of it. Except that for years, like the last 20 years at least, people have been trying to simplify it, to draw it in a way that we can understand it. So this is what you have on the cartoon on the left. So basically you have primary production at surface that is taking up CO2, that is getting into different interaction in the organisms that are part of the ocean surface. These interactions generate aggregates. These aggregates have different interaction with the plankton community that is around and the microbial processes that are driven the formation and degradation of these aggregates, and then they sink to depths where they sequester carbon. So when you put it that way, it's very simple, but like I said, it's a lot of biological processes that are involved in the shaping of the different fluxes that you have around the process. So, sorry. The way it's been studied for years, it's by trying to measure the flux at depth. So people have been deploying sediment trap for more than 50 years now, collecting particles at depth and trying to infer flux and the change of flux with depth. This is what you have on that scheme here, where you put the sediment trap, you collect the material, and then you analyze it for its chemical and sometimes the biological component of it. The thing is with sediment trap, it's really time consuming, integrate space and time, and it's costly to deploy. I'm not saying that you should give up on using sediment trap because it's still a standard tool in the oceanography and biogeochemistry, except that today we have other tools that can be used to assess fluxes or assess the function that affecting the fluxes. If you put everything that has been done for the last 60 years on a map, this is what it actually looked like. So this is a drawing from Onjo that did a compilation of every fluxes that were available since the 60s, pretty much. And so it looks pretty good, actually. You have a good coverage of fluxes around the global ocean, except that when you put that in perspective of whatever has been done on the impact of the moon on the Earth, that's what you have in the black dot bar here, compared to what we know on the deep ocean, including the sediment trap measurement, it's the blue bar. There's an order of magnitude difference between the two. And so this, to me, it's just a simple drawing that I did in like about 10 years ago, but it's still accurate. It's based on a search in the bibliography. And it tells you really that we don't know much about the deep ocean. We don't know much about the fluxes itself, just because it's hard to measure, but also because the tools for high throughput measurement kind of were not available at the time. At the same time, we've been looking at whatever is shaping the flux, so whatever is happening at the surface and driving export of material from the surface. This is the same thing. For years, we've been looking at a partial view of the ecosystem. It's like looking through a keyhole. We're focusing on key organisms, a few that could be measured or could be sampled. And so with the area of the omics or the imaging, now we can get into the depths of the complexity of the community at surface that is driving the flux. So that gives a complete new perspective about how to study the biological carbon pumps. This is just an example because it can be true for any kind of biogeochemical cycle. So when I'm talking about the new data that arose from the last 10 years, I'm mostly talking about the omics, whether it's metabarcoding data, genomics, transcriptomic data. We had a great example before from Corday about the protein that are now available. So it's that field is growing exponentially, and there is a lot of things that can be done, except that I mean, it takes time to see how to process the data in a way that can be used for the process that we've been studying for years. And in parallel, there are the imaging data. It's pretty much the same. In our lab, we use much more imaging data than genomics, and I'll show you few examples of what we can do with the images. And I like to try to use both at the same time, because basically the genomic data, most of the time they give you a semi-quantitative view of the community that is present. It's a semi-quantitative data. Whether the imaging data, they can be completely quantitative. And so when you have the two views at the same time, sometimes it gives you a similar view of the system. Sometimes you have a complementary view of the system. So just to zoom in a bit into the imaging data, it's been quite challenging because when you look at a plankton community, it goes from very tiny cell under the micrometer, if you go to viruses, it's even less, to something that can be large as few tens of centimeters for the larger karyotes. And so if you put that into our scale, the human scale, it would be like trying to capture at the same time something that is as large as a bee or of a bee and the largest mountain on Earth. So it tells you basically with that kind of perspective that you can't study the full ecosystem using only one instrument. So what we've been doing for years is trying to combine the instrument, except that they have their own biases and their own optical characteristic, which is pretty challenging then to put back together to have a complete view of the ecosystem. But because of the advances of machine learning and imaging tools that have been developed for the last 15 years now, there is a common framework to plug all these measurements into a shared software online that we develop, Ecotaxa, to try to analyze the plankton community. I mean, again, I'm going from micro scale to centimeter scale at the same time. This software we've been developing it because it was basically too challenging to try to manage imaging data on our own. And this is a community tool. You can collect data with any type of imaging instrument, standardize them, plug it there, and use machine learning to speed up the analysis and the annotation of these images. You can annotate the image, but you also can use, which is actually much easier, use traits. So you can use the size of the organism, the grayscale of this organism, any type of measurement that you can do on them, which is very important because the amount of data that we have now, it's just to give you the scale, it's 500 million images that we have to analyze to be able to understand things at global scale. So clearly, it's impossible to annotate everything. This is the example of what we have and that you can use. So it's called Ecotaxa, and you can go on it, browse, request access to different projects, and you'll see a bunch of images for specific crews, specific location. There is a right associated to the images, depending on whether they're open, private, whatever. And you can do them. And on the side of them, we develop an echo part, which is related to another instrument, the optical instrument that allow us to measure fluxes. So I'm mentioning this because when you go back to the question of the beginning, the plankton, the biological carbon pump, you have to have the two. You have to measure the fluxes and you have to know what are the organisms that are present. So you can do it by omics or you can do it by images. And so we've done that in 2016 with Damien Veillard and others, where we took the problem in a different way. We were saying like, okay, we don't understand who is there, what they are doing, but we're going to try to put everything on the filter, put that in a mixer kind of, and try to understand what are the organisms that are there and potentially influencing carbon fluxes at different depths. So we did that by collecting all the data that were the samples that were collected during the Tara Ocean cruises expedition, and then we analyzed the community to understand who was there, how much the flux was potentially evolving between depending on the community that was present, and how much so how the organism were doing it. With what type of processes and how much carbon was sequestered. So the way we did that is by trying to put all this data together. So for that, we needed to use an abstraction, trying to put all this organism in a single framework. So this is a more recent paper from Samuel that was part of the paper as well in 2016. But what he did, it developed an interactome. An interactome is just a co-occurrence network of all the species that were present or absent at a given station. So that's all the station that we sampled. And so you can look at the co-occurrence pattern of these different species. This is a mathematical tool. Then you can use that tool to explore whatever relationship there is with environmental condition. These plots at the bottom, it's just showing that basically the topology, so the shape of that network, is really driven by environmental conditions. So when you do that, that's what we did in that paper in 2016. We were trying to correlate. So this is the map of the flux at the different location. And then we had a bunch of species that were present from a prokaryote to viruses to eukaryote. And so we were trying to correlate not only the individual species, but the network of species that were corresponding to the flux at different depths. And so that's what you have on that plot. It's a sub-part of that network that is totally, so all these species, because they're part of a sub-network of the total network, they're correlated at the same time at the variation of the flux. But then you can explode that sub-network and try to look at their individual correlation to the flux, which would be the same thing as doing, for example, multilinear regression, and look at how central these species were in that subnetwork. Meaning that if they're very at the center of the network, they're very important for the structure of the ecosystem. If you remove it, basically the subnetwork doesn't exist anymore. So I give you two ideas at the same time. The potential interest of that species to modulate fluxes, but also the potential interest of that species for the surrounding species. So we did that for the eukaryote, we did that for the prokaryote, we did that for viruses. It was three independent network, and then we could rebuild the full network of potential species associated to the biological carbon pump. It was the first time that it was done, but when we did that, at first there was one thing that popped up for the eukaryote. There was another story for viruses. I'm not going to talk about it today, but so it was collodarian. Collodarian were highlighted in that study by statistical analysis as potential interest for making a strong variation of fluxes at the different location. There was only one paper at the time that suggested that collodarian were species of interest for the biological carbon plump from Rampit, based on a time series of the coast of the UK. And so we were wondering if it was just because of an amplification issue based on the genomic data, or if it was actually true. So luckily, we had imaging data. And so we dove into the imaging data and tried to estimate the total abundance of collodarian, but in general, rhizarians, because they're not disturbed by in situ imaging instruments compared to whatever you can do with nets. And we were trying to understand what was the pattern of their distribution, just the total abundance of that species. And what we found, going back to more than 3,000 profiles that we had with the same instrument, is that when you get between like 30 north and 30 south, in yellow, what you have here is the distribution of the abundance of the total biomass in carbon of copepods compared to radiolarians. And so actually they were very abundant and sometimes even more abundant in terms of biomass, carbon biomass than copepods. So that was an independent validation of a signal that we find correlating just genes presence with fluxes that was validated by an independent measurement based on an imaging data set. So it was the first time that we put that as an evidence, and then Tristan, who led that study, wanted with like a few colleagues to investigate that a bit more. And so having correlation at given station was a good start, but then, so we talked a lot about machine learning before, thanks to Corday that present a lot of different type of algorithms. And so we did this similar things, trying to predict the global distribution of that of the different collodarians that were present. So Laetitia that led that study, she took all the different observation of rhizarian that we had and other imaging, copepods. She built an habitat model, so it's some sort of a species distribution model, using boosted regression tree, and try to explain the pattern of the distribution of the different species. And what she found is like a similar pattern. Obviously, it's based on the same kind of data, with basically a large, significant abundance of here is just a solitary collodarians in mid-latitude compared to copepod. And so when you look at the total biomass, they actually in the same range for the equatorial region. But then the beauty of the imaging data is that you can go in depth into the images. And so Manon, which was a PhD student from Tristan, went into the full data set, and she spent a lot of time validating what was called rhizarians in different groups. So she went through the images, trained an algorithm to do a fast sorting, and then validate everything. So she could sort all the in that category, all radiolarians, foraminifera, feodarians, and rhizarians. And so then she could investigate the pattern of the distribution of these different groups inside that family. She coupled that to a small physiological model and tried to explain the variation of the contribution of Phaeodarian, which were highlighted as the most abundant of this category present in the imaging data. And so she could find actually that about 10% of the flux attenuation, so actually the flux that is captured by these organisms, could be explained by only Phaeodarian that were observed at depth. She also looked at the carbon demand based on that physiological model of this organism and could explain a large variation also of the demand of carbon at depths linked to these organisms. Then she investigated she looked at the biogenic silica biomass that was generated because of the presence of this organism, because it's the only source, Phaeodarian at that depth are the only source of potential silica. And so she could map the contribution of the different species to the total silica biomass that was measured at depth. So it's just to give you an example of how you can move from single observation to something that get into mechanistic understanding of the variation of different fluxes. So so I talk a lot about fluxes, but the beginning of what is actually sinking, it's what is produced at surface. So with a postdoc, Alexandre, we try to look at carbon fixation based on metagenomic data. So we had a catalog of mags, and we look at the potential pathways that could impact carbon fixation at surface at about 200 locations, because that's the amount of station that we had. So I'm not going to get into the detail, but carbon fixation can be done by different pathways, the C4, C3, etc. And what we did is we used, again, machine learning algorithm to predict the distribution of different metabolites and pathways of the carbon fixation using some sort of, a species distribution model. Except that when you look at a metabolic pathway, you have connections of enzyme in that pathway. So you need to take that into account. You don't want to predict individual enzymes or metabolites. You want to predict the distribution of a total pathway. So we used for that something that came up when we did that paper. I think the method came up in 2022, and we used in that paper in 2024. Which is called the multivariate boosted tree regression. What the method does, it actually consider that two here, for example, on the yellow pathway there, on the orange pathway there, or the dark one, the black one, they have to be predicted at the same time. So depending on the expression pattern that you have at the different location, you need to take that into account when you do the prediction. I'm not getting into the detail here. If you want, you can go to have a look at the paper. But again, that's an example of how to use machine learning to not only predict the species from surface or whatever, but also the function that are related to this species. Because we use the MAGS catalog, then we could also investigate not only the function, but what was the species leading to this that expressed this function. And we actually found that there was not a specific aspect of the function. There was a lot of redundancy in the prediction of the metabolic pathway at the global scale for the carbon fixation. I'll skip that, but Alex developed then, because of that, he wanted to develop a common framework to do this type of prediction. And so there is a tool that is available that is called Cephalopod that allow you to do species model distribution, but using different type of algorithm, find the best one and make your map at global scale on whatever data you want to use, whether it's presence only, continuous data, or proportion. So it really fits well what imaging data, but also genomics ones. So all that was correlation, even though we tried to understand a bit the mechanistic under it. And so with Damien, for years we've been trying to link this to understand or at least put information on correlation, and then move from correlation to mechanistic understanding of the biological carbon pump. So what this means is that we want to link a very fine understanding of cells. That's a gene scale model, for example, of Prochlorococcus. And we want to link that to something that is global. And so that's another type of model. So you had a seminar last week about biogeochemical modeling. So that fits the last week's seminar, and that's going to fit next week's seminar. And so what we want to do is to link the two and use highly defined information of the biology to inform biogeochemical processes at global scale. So Antoine, which was a PhD student from Damien, worked with us, and that's what he did. So he merged the two to try to understand, to try to model the biology component of the ocean and its implication for the biogeochemistry at global scale. So he embedded a genome scale model into an Earth system model. So that's one of the outcomes. I'll skipping the detail here, but you have a lot of information in the paper that is in bioRxiv, and it will be published in the coming months. So what you have, well, that's the mechanic under it, but what you can see is that here is the Earth system simulation of the growth rate of picoeukaryotes, and what you have here is the GSM model simulation of the growth rate of Prochlorococcus. And what you see is that the two patterns are very similar. If you look at latitudinal transect here, you have very similar behavior, whatever the depths of the organisms are. You have a mismatch, but the mismatch can be explained because this is just one subfraction of the total picoeukaryote. The good thing of doing this is that then you can move into the detail of the biology. So you can try to understand what are the co-limitations of this organism at different locations based on the biogeochemical output of the ESM, or you can even predict whatever metabolite the organism is producing at different locations depending on the environmental conditions. And so that really, to me, fits the goal of BioGeoSCAPE, and where we want to go. It's really to understand the fine biology that is driving biogeochemical fluxes at global scale. And on that, I will stop. I have a lot of people to thank, PhD student and postdoc from the lab, senior PI that I'm working on a daily basis, and people that are generating data and software that we use also to analyze the global data set. And Damien, with whom I've been spending for the last maybe 50 years now, thinking about how to move a bit forward to understand the biology of carbon pump.
Wonderful. Thank you, Corday and Lionel. Those were both fantastic talks. We have a short time left, but I'm hoping we can squeeze in at least a couple of questions. So I just want to kick off with a question here for Corday from Holly, who essentially was asking about how much of the phylogenetic information could be taken into account when thinking about how to model the metal cofactors and the proteins.
Yeah, so relatedness, both in sequence and in structure, it is really at the heart of all of these models. So it depends a little on what you mean by phylogenetic, right, in this context. But if you mean sequence similarity, then yes, sequence similarity is a major piece of the prediction of all of these models. With some of them, like AlphaFold, there's a more direct reliance on phylogenetically related proteins and sequences, right? So I think I showed in that schematic of AlphaFold, part of its initial step is to take not only the input sequence that you've given it, right, but then also to search all of these sequence databases, the PDB, to look for related sequences and structures. And then it builds sort of a more general representation of your sequence and potential structures related to your sequence based on that, right? So it's very directly using similar phylogenetically similar proteins. Some of the others, it's a little bit more abstracted, but it is there, right? The problem is that structural, right, sort of the structural similarity of two proteins is not necessarily always directly tied to the sequence similarity, right? You can have two very different sequences that bring you to a structure that's quite similar. So that's why, you know, having both, I think, gets you to a better answer.
Perfect. I have a question now for Lionel from Jacob Cram. He says, you showed in your 2016 paper that colidaria have high variable importance when related to flux and are central to network related to flux. So it's a two-part question. The first part is, are they positively or negatively related to flux, and what do you see as potential mechanisms for the relationship? And then the second part asks, what does the centrality in the interaction network tell us about colidaria's ecology and biogeochemistry? There's a lot there.
Yeah. So thanks for the great question. I mean, I'm not a biologist, so Tristan would have been here and could have talked maybe like an hour just about the biology of the Hesarian. The thing that we've been chatting about is that, so the correlation, I think if I recall, it was a positive correlation with flux. And so it's also because they thought to be a flux feeder. So they just capture whatever is sinking. So they don't contribute to the flux directly, but they contribute to the attenuation potentially of the flux. But it's a positive correlation. And in terms of the centrality, it's true that they are very central in the network. It's not the most central one, but it means that they hold the community together. It can be because they're just very abundant, so they are there, and so whatever is around is living around them. So it's a statistical relationship more than a mechanistic one. And for the mechanistic, I really don't know. I'm not into, like I said, I'm not really a biologist, so I have a hard time to explain potential interactions led to these organisms.
I'm just gonna throw in a quick follow-on question, although we're running out of time. But Lionel, what you just said about the interactions made me think about how we could use these models to explore those interactions more. And I'm guessing that's something we could probably talk about at the workshop in Woods Hole.
Yep.
Well, we're at 5 o'clock. Well, 5 o'clock for me. Other times for other people, we're on the hour. So I want to thank Lionel and Corday again. Those were both brilliant talks. So many questions that we could ask, but we don't have time. Thank you, everyone, for taking part and for attending. And as Christoph said, these talks will be posted online. So if you miss part of it or if you want to watch it again, you can.
See everyone next week.